Every cell in your body is the scene of an ancient crime. Right now, as you listen to this, trillions of tiny structures inside your cells are working, converting the food you ate this morning into the energy that keeps your heart beating, your lungs breathing, your brain making sense of these very words. We call them mitochondria. You were taught in school that they're the powerhouses of your cells. That's true, but it's not the whole truth. Those mitochondria, every single one of them, carry their own DNA. Not your DNA, theirs. It's circular, like a bacterial chromosome, coiled in a tight loop of 16,569 base pairs. It uses a slightly different genetic code than the rest of your genome. It divides on its own schedule by binary fission, the same way bacteria divide, and it is passed down exclusively through your mother's line, an unbroken chain of maternal inheritance stretching back across unfathomable time. This is strange. This should not be the case. If mitochondria were simply parts of your cells, components built by your cells from your own genetic instructions, the way your ribosomes are built or your cell membrane is assembled, they wouldn't need their own DNA at all. And yet there it is, a separate genome, a separate heritage, a separate evolutionary history woven into the fabric of every complex cell on Earth. Your heart muscle cells contain so many mitochondria that the organelles fill nearly a third of each cell's volume. A human egg cell, the largest cell your body produces, can harbor between 100,000 and 600,000 mitochondria, each carrying its own circular chromosome. Collectively, your mitochondria produce roughly your own body weight in ATP every single day. That molecule, adenosine triphosphate, is the universal fuel of life. And virtually all of it comes from structures that are not, in the deepest evolutionary sense, you. So what are they? 2 billion years ago, longer than any animal has existed on this planet, longer than the oldest fossil you've ever seen in any museum, longer, in fact, than the entire span of visible complex life, a cell swallowed another cell. It should have been digested. It should have been lunch. Instead, something happened that had never happened before and would never need to happen again in quite the same way. The swallowed cell survived. And over vast stretches of time, the two organisms, predator and prey, host and captive, became so dependent upon each other that neither could survive alone. That merger produced the first complex cell, the ancestor of every plant, every animal, every fungus, every protist on Earth. Every redwood that has ever reached toward sunlight, every whale that has ever breached the ocean's surface, every mushroom, every fern. You. And for most of the 20th century, only one scientist truly understood what had happened. Her name was Lynn Margulis. She published her evidence. She laid out her arguments with rigor and precision. And the scientific establishment laughed. This is the story of the most important partnership in the history of life on this planet. It is the story of how we came to exist, not through competition alone, but through an act of cooperation so profound it redefined what it means to be a living thing. And it is the story of a woman who refused to be wrong quietly. To understand what happened 2 billion years ago, we need to understand what came before. And what came before was, by any measure we might use, astonishingly simple. For the first billion years of life on Earth, the only living things were prokaryotes, bacteria and archaea, single cells without a nucleus, without internal compartments, without the elaborate architecture we associate with the cells of our own bodies. They had no mitochondria, no endoplasmic reticulum, no Golgi apparatus, no organelles at all in the way we usually understand the term. Just a membrane, some ribosomes, a chromosome, and an extraordinary capacity for chemical innovation. These prokaryotes were extraordinarily successful. They colonized every conceivable environment, boiling hot springs where water reaches temperatures that would cook flesh in seconds, frozen tundra, the deepest ocean trenches where pressure would crush a submarine, the interiors of rocks kilometers below the Earth's surface. They invented photosynthesis, not once, but multiple times, in different ways, using different wavelengths of light and different electron donors. They learned to fix nitrogen from the atmosphere, making it available for biological use. They developed metabolic pathways of stunning chemical sophistication, extracting energy from hydrogen, from sulfur, from iron, from compounds that most living things today would find utterly useless. For 1 billion years, they were all there was. And in many ways, they were enough. But they had a limitation, a ceiling they could not break through. And the reason for that ceiling is one of the most elegant constraints in all of biology. Prokaryotic cells generate their energy across their outer membrane. The proteins of their electron transport chains, the molecular machinery that converts food into usable energy, are embedded in the cell's surface. This means their energy production scales with their surface area. And here is the problem. As a cell gets larger, its volume increases faster than its surface area. This is the cube-square law. Double the diameter of a sphere, and you get 4 times the surface area, but 8 times the volume. A bacterium that doubles in size doesn't get twice as much energy. It gets less energy per unit of volume. The math is unforgiving. So prokaryotes stayed small. They had to. There was no other option available to them, no workaround, no evolutionary trick that could break this fundamental geometric constraint. The largest bacteria ever found are still visible only under a microscope. The smallest are barely more than molecular assemblages. And within that narrow range of sizes, there is a hard upper bound on complexity. You cannot build a brain from bacteria. You cannot build a tree. You cannot build a mushroom, or a coral reef, or a hummingbird, or a beating heart. Not because bacteria lack sophistication. They are, as we've said, masterful chemists, capable of metabolic feats that eukaryotic cells cannot match. But because complexity costs energy. Internal membranes cost energy. A nucleus costs energy. A cytoskeleton costs energy. Multicellularity, with its specialized tissues and coordinated development, costs energy most of all. And prokaryotes simply could not afford it. For 2 billion years, 2 billion, this was the state of life on Earth. Microbial mats and stromatolites, films of bacteria in shallow seas, archaea thriving in extreme environments, vast invisible ecosystems of chemical cycling and molecular exchange. But no eyes, no movement beyond the microscopic, no predation, no thought. If you could have stood on the shore of that ancient Earth, an impossibility since nothing with legs existed to stand, you would have seen a world that looked almost lifeless. A pale sky, a quiet ocean, its surface unbroken by any fin or fluke. Perhaps a faint sheen of green or purple in the shallows where microbial mats caught the light. and silence. A silence lasting longer than the entire age of animals. The planet breathed, but only chemically. It hummed, but only at the molecular scale. Life was everywhere, and it was invisible. Then the world changed, and it changed because of poison. Around 2.5 billion years ago, a group of prokaryotes called cyanobacteria, those early masters of oxygenic photosynthesis, had been pumping oxygen into the atmosphere as a waste product of their metabolism for hundreds of millions of years. At first, the oxygen didn't accumulate. It reacted with dissolved iron in the oceans, forming the vast banded iron formations that today supply 60% of the world's iron ore. It reacted with gases in the atmosphere. It was absorbed by minerals in the Earth's crust. The planet's chemistry soaked it up as fast as the cyanobacteria could produce it. But the cyanobacteria were relentless, and eventually, around 2.4 billion years ago, production outpaced absorption. Oxygen began to accumulate in the atmosphere for the first time. To the organisms that dominated Earth at the time, oxygen was toxic. It was corrosive. Reactive oxygen species tore apart the delicate molecular machinery of anaerobic life, shredding proteins, damaging DNA, disrupting the metabolic pathways that had sustained life for 1 billion years. The cyanobacteria were poisoning their own world. Not intentionally, not even consciously, just inevitably. We call it the Great Oxidation Event. It was, in effect, the first mass extinction, caused not by an asteroid, not by a volcanic eruption, but by life itself. The event was so catastrophic that oxygen reacted with atmospheric methane, the greenhouse gas that had been keeping the planet warm, triggering the Huronian glaciation, a global freeze so severe that ice may have reached the equator. And for a time it must have seemed like an ending. The warm oceans cooling, then freezing, the iron in the seas rusting red before sinking to the seafloor in great, silent layers. The methane haze that had softened the sky for 1 billion years, thinning to nothing, revealing a hard, cold sun. The organisms that had owned the Earth for longer than we can meaningfully comprehend, retreating to cracks, to vents, to the last warm pockets of an anoxic world. Some went extinct entirely. The planet they had known was gone. But oxygen, for all its destructive power, had a remarkable property. If you could learn to use it, if you could harness its reactivity instead of being destroyed by it, you could extract far more energy from food than any anaerobic metabolism allowed. Roughly 18 times more ATP per molecule of glucose. It was, in metabolic terms, a revolution. Somewhere in this newly oxygenated world, a particular kind of bacterium figured this out. An alphaproteobacterium, a relative of the organisms that today cause typhus and Rocky Mountain spotted fever, evolved the molecular machinery to use oxygen as the final electron acceptor in its respiratory chain. It was small, it was efficient, and it was very, very good at what it did. That bacterium is the ancestor of every mitochondrion in your body. And it was about to meet the other half of its future. Now we come to the moment, the event itself. We don't know exactly when it happened. We don't know where. We don't know the precise circumstances, whether it was a shallow sea or a tidal flat or a hydrothermal vent or somewhere else entirely. but molecular evidence has given us increasingly sharp portraits of the two cells involved. And the story those portraits tell is remarkable. One partner was an archaeon. Not just any archaeon, a member of a group we now call the Asgard archaea. And the discovery of these organisms is itself one of the great detective stories of 21st century biology. In 2010, a team led by Theis Ettema at Uppsala University in Sweden began analyzing sediment samples dredged from the bottom of the Arctic Ocean near a hydrothermal vent field between Norway and Greenland called Loki's Castle. Named after the Norse trickster god, the site sits along the mid-Atlantic ridge, where tectonic plates pull apart and superheated water erupts from the ocean floor. The sediments contained DNA from organisms no one had ever seen before. When the genomes were pieced together from fragments, a painstaking process called metagenomics, the results, published in 2015, stunned the scientific world. These archaea, which the team called Loki archaeota, carried genes that had previously been found only in eukaryotic cells. Genes for building protein scaffolding, genes for membrane remodeling, genes for the kind of complex internal structures that prokaryotes weren't supposed to possess. More Asgard archaea followed: Thor archaeota, Odin archaeota, Heimdall archaeota, each named for a Norse deity, each carrying more eukaryotic signature proteins. A 2025 study in Nature confirmed what many had suspected. The Asgard archaea contributed the dominant share of genes to the ancestor of all complex life. The majority of core eukaryotic cellular systems, the machinery for managing the genome, building internal structures, processing information, traces back to Asgard ancestry. And in 2019, after 12 years of painstaking laboratory work, Japanese microbiologist Hiroyuki Imachi and his team achieved what many thought impossible. They cultured a living Asgard archaeon. They called it Prometheoarchaeum syntrophicum. It grew agonizingly slowly, doubling its population every 14 to 25 days, compared to 20 minutes for common laboratory bacteria. But when they finally observed it, what they saw was suggestive. The organism extended long, branching, tentacle-like protrusions from its cell body, and it seemed to form intimate physical associations with other microbes, pulling them close. Perhaps, and this is speculation, but informed speculation, we were looking at an echo of that ancient partnership, an archaeon reaching out physically to other cells, drawing them near. So picture this ancient Asgard archaeon. 2 billion years ago. It was larger than most prokaryotes, perhaps already possessing rudimentary internal structures. It lived in a world newly flooded with oxygen, a gas it could not use, a poison it had to endure. And near it, perhaps feeding on the same organic matter, perhaps consuming the archaeon's metabolic waste products, was a small alphaproteobacterium that thrived on oxygen, that breathed it, that turned it into energy with an efficiency nothing else on Earth could match. What happened next remains the subject of ongoing scientific debate. Perhaps the archaeon tried to eat the bacterium, phagocytosis or something like it, and failed to digest its meal. Perhaps the bacterium invaded the archaeon as a parasite, boring through the membrane the way infectious bacteria do today. Perhaps they were already living in close association, a syntrophic relationship, each feeding on the other's waste products, hydrogen flowing one way and organic acids flowing back. and one simply slipped inside the other. The hydrogen hypothesis, proposed by William Martin and Miklos Müller in 1998, suggests exactly this kind of metabolic codependence as the starting point. However it began, the result was the same. The bacterium ended up inside the archaeon, and it did not die. This is not as extraordinary as it sounds. Cells engulf other cells all the time. Bacteria invade hosts constantly. Parasites take up residence inside other organisms as a matter of routine. What is extraordinary? What makes this event singular in the entire history of life on Earth is what happened next. Nothing dramatic. For a very, very long time. The bacterium lived inside the archaeon. It continued to respirate. It continued to use oxygen to generate ATP. And the archaeon, for the first time in its lineage's history, had access to aerobic energy. Not much at first, a trickle, but a trickle of something the outside world had made newly valuable. Over hundreds of millions of years, and this is the part that is hardest to grasp, the timescale over which this partnership was forged, the two organisms became increasingly dependent on each other. The bacterium lost genes it no longer needed because the host cell's environment provided what those genes had once encoded. Genes for building its own cell wall, genes for synthesizing amino acids the host already supplied. One by one, these became redundant, and mutations that disabled them were no longer punished by natural selection. Meanwhile, something even more remarkable was happening. Genes were migrating from the bacterium's genome into the host's nucleus. Wholesale transfer of genetic information, the bacterium's own DNA, copied and integrated into the archaeon's chromosomes. We can still detect this today because fragments of mitochondrial DNA sequences are scattered throughout your nuclear genome, like fossils embedded in sedimentary rock. A recent study found that most humans carry detectable mitochondrial DNA insertions in their nuclear chromosomes. The bacterium shrank. It simplified. Over vast timescales, it lost its independence, gene by gene. It became something that was no longer quite a bacterium, but not yet what we would recognize as a mitochondrion. It became an obligate endosymbiont, an organism that could not survive outside its host, and its host could not survive without it. There is no word in any human language for what these two organisms were to each other during that long transition. They were not predator and prey, not parasite and host, not parent and child. They were something older than any of those categories. Two separate living things in the slow, patient process of becoming one. The bacterium was losing itself. The archaeon was gaining something it could never have built. And neither of them chose it. It simply happened. The way rivers carve canyons, the way gravity draws moons into orbit. Slowly, silently, irreversibly. Two became one. And that one was something the world had never seen before. A eukaryotic cell. Why did this matter? Why was this merger, out of all the trillions upon trillions of interactions between cells across billions of years, the one that changed everything? The answer comes down to energy, and specifically to the geometry of energy production. Remember the problem that had kept prokaryotes small for 2 billion years. Energy generation happens at the membrane. Surface area limits power. Volume determines demand. The cube-square law creates an impassable ceiling. A bacterium cannot grow large without starving itself. But a cell with mitochondria inside it has shattered that constraint. It doesn't generate energy at its outer surface anymore. It generates energy at the membranes of its mitochondria. Internal membranes that can be folded, crimped, stacked, multiplied. The inner membrane of a mitochondrion is elaborately convoluted into structures called cristae, folds that vastly increase the surface area available for the electron transport chain. A single mitochondrion can have hundreds of these folds, and a single cell can contain hundreds or thousands of mitochondria. The numbers are staggering. An average human cell contains roughly 100 mitochondria. But that's an average. It conceals enormous variation. A liver cell, which processes toxins and manages metabolism around the clock, contains between 1 and 2,000 mitochondria. A cardiac muscle cell, the kind that keeps your heart beating, is so densely packed with mitochondria that they occupy up to 30% of the cell's total volume. And a human egg cell, the largest cell your body produces, can harbor between 100,000 and 600,000 mitochondria. So many that mitochondrial DNA accounts for up to 90% of the egg's total DNA content. This changes everything. A eukaryotic cell can be 10,000 to 100,000 times larger by volume than a typical bacterium, and it can power that volume because its energy production scales with the number of internal organelles, not with its outer membrane. Nick Lane, a biochemist at University College London who has written extensively about the energetics of eukaryogenesis, has estimated that the energy budget of a eukaryotic cell is roughly 200,000 times greater per gene than that of a comparable prokaryote. 200,000 times. That is not an incremental improvement. That is a revolution. With that energy, a cell can afford complexity. It can build a nucleus, a membrane-bound compartment to protect and organize its expanding genome. It can develop an internal skeleton, a cytoskeleton of actin and tubulin and intermediate filaments to maintain its shape, transport materials, and divide with precision. It can create the endoplasmic reticulum, the Golgi apparatus, lysosomes, peroxisomes, an entire internal economy of specialized compartments. It can evolve true phagocytosis, the ability to engulf large particles, including other cells, opening the door to predation, to food webs, to ecosystems of astonishing intricacy. And, crucially, it can afford to cooperate with other eukaryotic cells. It can afford to become multicellular. This is the part that doesn't get enough attention. Multicellularity has evolved independently dozens of times in the history of life. In animals, in plants, in fungi, in red algae, green algae, brown algae, in slime molds, in at least one group of amoebae. Every single time, without exception, it has evolved in eukaryotes, never in prokaryotes. Not once, not ever. Bacteria form colonies. They form biofilms, sophisticated communities with chemical communication and division of labor. They form mats that can carpet the ocean floor, but they do not form true multicellular organisms with differentiated cell types, specialized tissues, developmental programs, and coordinated organ systems. The energy cost is simply too high. They cannot afford it. Every oak tree, every elephant, every coral reef, every jellyfish, Every human being who has ever drawn breath exists because of that ancient merger. Because one cell swallowed another and kept it alive. The entire visible biosphere, everything you can see when you look out a window, everything you have ever touched, everything that has ever looked back at you with eyes, is the downstream consequence of a single event of endosymbiosis. Think about that for a moment. Every color you have ever seen was seen with eyes that only exist because of that merger. Every sound you have ever heard was heard by ears whose cells are powered by the descendants of that bacterium. Every thought you have ever had, every memory, every flash of recognition, every moment of wonder, including this one, was computed by neurons burning fuel that mitochondria provided. The merger didn't just make complex life possible, it made experience possible. It made knowing possible. It happened once. We have no evidence that mitochondrial endosymbiosis ever occurred independently a second time. One event, one partnership, and from it, all complex life on Earth. Lynn Petra Alexander was born in Chicago on March 5, 1938. She was, by all accounts, brilliant and difficult in roughly equal measure. Which is to say, she was the kind of person who changes the world and infuriates everyone in the process. She entered the University of Chicago at 14. Not a typo. 14. The university had an early entrance program for gifted students, and young Lynn was gifted in ways that her instructors would remember decades later. She earned her bachelor's degree at 19, her master's in genetics and zoology from the University of Wisconsin at 22, her PhD in genetics from the University of California, Berkeley in 1965 at 27. By then, she had already been married to and divorced from Carl Sagan. Yes, that Carl Sagan, the man who would go on to write Cosmos and bring astronomy to millions. They met at the University of Chicago when she was a precocious undergraduate and he was a graduate student in physics. They had 2 sons, Dorian and Jeremy. The marriage ended in 1964. She would later remark, with characteristic bluntness, that she'd had great taste in husbands, but very little patience for marriage. In her second marriage, to crystallographer Thomas Margulis, she took the name by which science would come to know her. In 1966, at 28 years old, a junior faculty member at Boston University with small children and no tenure and very little institutional support, Lynn Margulis sat down and wrote a 50-page paper that would upend our understanding of how complex life evolved on Earth. The paper was called On the Origin of Mitosing Cells. In it, Margulis argued, with a density of evidence and a breadth of reference that astonished even her critics, that the eukaryotic cell was not a single organism that had gradually evolved greater complexity over time. It was a chimera, a fusion, a symbiotic union of separate prokaryotic cells that had merged, become interdependent, and eventually fused into something new. Mitochondria, she argued, were once free-living alphaproteobacteria. Chloroplasts, the organelles that perform photosynthesis in plant cells, were once free-living cyanobacteria. She even argued, more controversially, and in a claim that modern science has since largely rejected, that the flagellum, the whip-like tail that propels many eukaryotic cells, might have originated as a symbiotic spirochete bacterium. She was not, technically, the first person to suggest these ideas. The Russian botanist Konstantin Merezhkovsky had proposed a symbiotic origin for chloroplasts as early as 1905. The American biologist Ivan Valin had extended the idea to mitochondria in the 1920s. But their ideas had been forgotten, or, more accurately, dismissed. By the 1960s, endosymbiosis was regarded as a quaint relic of early 20th century speculation, the scientific equivalent of phlogiston theory or the luminiferous ether. The consensus held that organelles had evolved gradually from within the cell through a process called autogenesis. To suggest otherwise marked you as ignorant of modern molecular biology. Margulis was not ignorant. Her paper was a tour de force that drew evidence from across the biological sciences: paleontology, comparative biochemistry, electron microscopy, the emerging field of molecular biology, genetics, and systematics. It laid out specific, testable predictions. If mitochondria were once bacteria, she argued, then they should contain their own DNA. And that DNA should resemble bacterial DNA more than it resembles the nuclear DNA of their host. They should have their own ribosomes, and those ribosomes should be more like bacterial ribosomes than eukaryotic ones. They should divide by binary fission. They should be sensitive to antibiotics that target bacteria. Every one of these predictions would eventually be confirmed. It was rejected by 15 scientific journals. The rejections came for a variety of reasons. Some editors felt the paper was too interdisciplinary. It crossed so many boundaries that it didn't fit neatly into any single journal's scope. Some reviewers simply disagreed. Some, as Margulis later recalled, didn't seem to have read past the first few pages. And some of the opposition was, let us be honest, not purely scientific. She was young. She was a woman. She was challenging the life's work of powerful men who sat on journal editorial boards and grant review panels. The National Science Foundation declared her findings totally unacceptable to important molecular biologists. The Journal of Theoretical Biology finally published it in 1967 under the name Lynn Sagan, since she was then still using her first husband's surname. It won the award for Best Faculty Publication of the Year at Boston University. Some of her colleagues in genetics and microbiology needed little convincing. But many researchers in other fields were deeply resistant. The paper sparked interest, debate, and, most of all, opposition. Margulis would spend the next 2 decades defending her ideas against criticism that ranged from thoughtful scientific disagreement to open mockery. Colleagues avoided her at conferences. Grant funding was sparse. She was the embodiment of the outsider scientist, not because she chose that role, but because the establishment pushed her into it. And she did not flinch. Not once. Years later, she told an interviewer, "I don't consider my ideas controversial. I consider them right." Science, when it works properly, doesn't care who's popular. It doesn't care about consensus or credentials or institutional prestige. It cares about evidence. It cares what's true. And the truth, and relentlessly, came down on Lynn Margulis's side. The first line of evidence was structural. Electron microscopes, growing ever more powerful through the 1960s and 1970s, revealed that mitochondria and chloroplasts are surrounded not by one membrane, but by 2. An outer membrane and an inner membrane, with a space between them. This is precisely what you would expect if one cell had engulfed another, the inner membrane being the original membrane of the captured bacterium, the outer membrane being the remnant of the food vacuole that swallowed it. A cell within a cell, still wrapped in the physical evidence of its capture. Then came the DNA. Through the 1960s and early 1970s, researchers confirmed that mitochondria contained their own genetic material, and that it was circular, like a bacterium's. Their ribosomes were smaller than those in the rest of the cell, closer in size and structure to bacterial ribosomes than to eukaryotic ones. They divided independently of the host cell by binary fission, the same mechanism bacteria use. They were sensitive to antibiotics like chloramphenicol and tetracycline, which target bacterial protein synthesis but leave eukaryotic ribosomes unaffected. They even had their own slightly different genetic code. The molecular words that translate DNA into protein are read differently inside the mitochondrion than in the rest of the cell. Each of these observations was consistent with Margulis's predictions. Individually, each one could have had alternative explanations. Together, they formed a pattern that was becoming very difficult to explain away. But the decisive evidence, the evidence that turned Margulis from heretic to prophet, came from a new kind of science: molecular phylogenetics. The ability to compare DNA and protein sequences across species and reconstruct evolutionary relationships from the molecular record. In 1978, Robert Schwartz and Margaret Dayhoff published a landmark study comparing the amino acid sequences of proteins from mitochondria with sequences from free-living bacteria. Their approach was simple in concept, revolutionary in implication. If mitochondria truly evolved from captured bacteria, their protein sequences should be more closely related to those of modern bacteria than to the nuclear-encoded proteins of their own host cells. If mitochondria evolved gradually from within the cell, their sequences should cluster with their hosts. The results were unambiguous. Mitochondrial proteins clustered with the alphaproteobacteria, a specific group of free-living bacteria, not with the nuclear genome, not with any archaeal lineage. The mitochondrian's family tree was bacterial. Its ancestry was bacterial. It was a captured organism, not a homegrown invention. Similar results followed for chloroplasts. Their genes nested neatly within the cyanobacteria on the tree of life, the same oxygen-producing photosynthesizers that had caused the Great Oxidation Event. Plants don't photosynthesize because they gradually evolved the molecular machinery to do so. They photosynthesize because their ancestors swallowed cyanobacteria and kept them. By the early 1980s, the accumulation of genetic, structural, and biochemical evidence was overwhelming. Mitochondria came from alphaproteobacteria. Chloroplasts came from cyanobacteria. The eukaryotic cell was, exactly as Margulis had argued in 1967, a symbiotic union, a community masquerading as an individual. In 1983, Lynn Margulis was elected to the National Academy of Sciences, the highest professional honor in American science. In 1999, President Bill Clinton awarded her the National Medal of Science. In 2008, the Linnean Society of London, the world's oldest active biological society, the institution that first presented Darwin and Wallace's theory of natural selection to the world in 1858, awarded her the Darwin-Wallace Medal. Only 4 women in history had ever received that medal at that time. And Richard Dawkins, the evolutionary biologist who had spent much of his career in intellectual opposition to Margulis on other questions, who had publicly and forcefully disagreed with her on the role of cooperation in evolution, wrote of her in 1995, "I greatly admire Lynn Margulis's sheer courage and stamina in sticking by the endosymbiosis theory and carrying it through from being an unorthodoxy to an orthodoxy. This is one of the great achievements of 20th century evolutionary biology, and I greatly admire her for it." From unorthodoxy to orthodoxy. It took 30 years. But the evidence didn't care about orthodoxy. It cared about truth. The story of endosymbiosis doesn't end with mitochondria, because the Great Merger happened again. Sometime between 1.5 billion and 1 billion years ago, the precise timing remains debated. A eukaryotic cell that already possessed mitochondria engulfed a cyanobacterium. And once again, instead of digestion, partnership. The cyanobacterium survived within its host. It continued to photosynthesize. Over time, it lost its independence, transferred many of its genes to the host's nucleus, and became the chloroplast, the organelle that converts sunlight and carbon dioxide into sugar and oxygen. With that second endosymbiosis, the eukaryotic world split into two great kingdoms of energy acquisition. Animals eat, plants photosynthesize. That fundamental division, the one that defines nearly every ecosystem on Earth, that separates the things that move toward food from the things that stand still and make it from sunlight, traces back to this second swallowing. Every salad you have ever eaten is a collection of cells powered by captured cyanobacteria. Every forest, every prairie, every field of wheat exists because of endosymbiosis. And it didn't stop there. Some eukaryotes swallowed other eukaryotes that already had chloroplasts, creating secondary and tertiary endosymbioses, layers within layers, partnerships nested inside partnerships. Brown algae, red algae, dinoflagellates, cryptophytes, each carrying inside them the molecular ghosts of multiple mergers, like Russian nesting dolls of ancient cooperation. In some of these organisms, you can still count the membranes. 2 from the original mitochondrial capture, 2 more from the chloroplast, and sometimes 2 more from the eukaryotic cell that swallowed the cell that already had chloroplasts. 6 membranes deep. 6 concentric boundaries, each one the scar of an ancient engulfment. Each one a wall that was once a border between 2 living things, and is now just an interior surface in a single cell. You could read them like tree rings, except what they record is not seasons, but mergers. Each ring a moment when one organism closed around another, and the 2 of them never let go. This is not what we were taught evolution looks like. The textbook picture is a tree, a branching tree, where lineages split and diverge, where species separate and go their own ways. Darwin's great insight. And it's real. It's true. Lineages do diverge. But endosymbiosis reveals something the tree model misses. Lineages also merge. Branches that split apart can come back together. Not through sex, not through hybridization in the conventional sense, but through the wholesale incorporation of one organism into another. The tree of life, it turns out, is not purely a tree. It is also a web, a network, a tangle of ancient mergers and partnerships that produced some of the most important innovations in the history of living things. Life did not take over the globe by combat alone. It took over by networking. Those are Lynn Margulis' words, and she was right. So what does all this mean? For you, specifically, personally, biologically. Consider this. Your body contains roughly 37 trillion cells. Every one of those cells, except your mature red blood cells, which have ejected their nuclei and most of their organelles to make room for hemoglobin, contains mitochondria. Anywhere from a handful, in the case of some white blood cells, to thousands in the case of your neurons, liver cells, and cardiac muscle. Each of those mitochondria carries its own genome, its own evolutionary heritage, its own ancestry separate from yours. You have two genomes, two family trees, two histories living inside the same body. Your nuclear DNA, the 23 pairs of chromosomes you inherited from both your parents, descends from those ancient Asgard archaea, modified by billions of years of evolution, the incorporation of thousands of bacterial genes, and the slow, relentless accumulation of mutations across incomprehensible spans of time. Your mitochondrial DNA, those 16,569 base pairs, those 37 genes, that tiny circular chromosome, descends from the alphaproteobacterium that was swallowed and never released. You got it from your mother. She got it from hers. Her mother got it from hers. An unbroken maternal line reaching back 2 billion years to the first cell that survived inside another cell and stayed. Every mother who has ever lived has passed her mitochondrial DNA to all her children. But only her daughters pass it to the next generation. The mitochondria in sperm cells are tagged for destruction after fertilization, actively eliminated by the egg cell in an autophagic process that begins within minutes of conception. Your father's mitochondria died with his sperm. Your mother's mitochondria live on in you. This maternal inheritance has given us extraordinary tools for tracing human history. Mitochondrial Eve, the most recent common matrilineal ancestor of all living humans, lived approximately 200,000 years ago in Africa. Every person alive today carries mitochondrial DNA descended from hers. Not because she was the only woman alive at the time, but because every other lineage of mitochondrial DNA has, through the lottery of having only daughters or only sons, or no children at all, gone extinct. Hers survived. Just as the original alphaproteobacterium survived. There is something almost sacred in that continuity, though it requires no faith, only chemistry. The mitochondria in your cells are not the same molecules that lived inside that first merged cell 2 billion years ago. They have been copied and divided and copied again billions upon billions of times across a chain of mothers so long it connects you to a world that had no oxygen in its sky and no name for anything. But the lineage is unbroken. The instructions have been passed hand to hand to hand, from a time before hands existed. You carry that inheritance in every cell. It is older than bone, older than blood, older than the idea of age itself. When mitochondria malfunction, the diseases they cause are devastating, and they preferentially strike the organs that need the most energy. Mitochondrial diseases can cause blindness, deafness, seizures, muscle weakness, heart failure, liver disease, and neurodegeneration. The mutation rate of mitochondrial DNA is 10 to 20 times higher than that of nuclear DNA, because mitochondria lack the sophisticated DNA repair machinery that protects the nucleus, and because their DNA sits in close proximity to the respiratory chain, the very machinery that generates reactive oxygen species as a byproduct of energy production. The ancient partnership is powerful, but it is not without cost. We think of ourselves as individuals, and in the way that matters to us emotionally, our consciousness, our sense of self, our experience of being a particular person in a particular time and place, that's valid. But biologically, the word individual is misleading. You are a consortium, a federation, a cooperative of two ancient lineages that merged so completely that they can no longer be separated. The eukaryotic cell is not an organism. It is a committee. and so are you. Lynn Margulis died on November 22, 2011, at her home in Amherst, Massachusetts, 5 days after suffering a hemorrhagic stroke. She was 73 years old. As she had wished, her ashes were scattered in her favorite research areas, near her home, returned, in a sense, to the microbial world she had spent her life studying. In the years since her death, the science she championed has not merely survived, it has exploded. The discovery of the Asgard archaea, first published in 2015, vindicated a key prediction of endosymbiotic theory, that the host cell in the original merger was an archaeon, not a bacterium. When Imachi and his team published their cultivation of the first living Asgard archaeon in 2020, the 12-year effort was hailed as one of the most significant microbiological achievements of the decade. The organism they grew, small, slow, tentacled, offered a tantalizing window into what the ancestral host cell might have been like. In 2025, a comprehensive study published in Nature by Tobiasen, Luo, Wolf, and Kuhnen analyzed the origins of every core eukaryotic gene traceable to the last eukaryotic common ancestor. Their results confirmed the dominant contribution of Asgard archaea. Most cellular machinery traces to archaeal ancestry, while the alphaproteobacterial endosymbiont contributed primarily the energy transformation systems. But the picture was more complicated than anyone in Margulis's time could have imagined. Genes flowed in from many bacterial sources, not just the mitochondrial ancestor. The eukaryotic genome is a mosaic, assembled from the genetic contributions of an entire ancient microbial ecosystem. And new endosymbioses are still being discovered. In 2024, researchers described what appears to be a nitrogen-fixing organelle, dubbed a nitroplast, found within a marine alga. The organism that became this nitroplast was, until recently in evolutionary terms, a free-living cyanobacterium. It appears to have crossed the threshold from symbiont to organelle within the last 100 million years or so, a blink in evolutionary time. Endosymbiosis is not just ancient history. It is an ongoing process. The mergers continue, right now, on this planet. In organisms we are only beginning to understand. Even Margulis's most controversial claim, that the eukaryotic flagellum originated from a symbiotic spirochete, remains unresolved rather than definitively refuted. The evidence has not supported it, but the broader principle it embodied, that symbiosis can produce structural novelty, not just metabolic novelty, continues to find support in unexpected places. And perhaps most remarkably, scientists have now begun to create endosymbioses in the laboratory. In 2024, a study published in Nature reported the successful implantation of bacteria into fungal cells, creating a novel, stable endosymbiotic relationship that persisted across generations. We are learning not just to observe the great mergers of the past, but to understand the principles that govern them, and potentially to engineer new ones. The story Margulis told is not finished. It may never be finished. Somewhere in an ocean we have not yet sampled, in a sediment we have not yet sequenced, in a cell we have not yet observed, the next merger may already be underway. Two organisms in the slow first days of learning to share a body. We would not notice. We never do. These things happen in silence, at scales too small for our senses, over timescales too long for our patience. But they happen. They are happening. There is a version of the story of life that goes like this. Existence is a war. survival of the fittest. Nature red in tooth and claw. Organisms compete. The strong survive, the weak perish. And that brutal calculus, repeated across 4 billion years, is the engine that built everything. That version is not wrong. Competition is real. Natural selection is real. Organisms do struggle, and that struggle does shape the trajectory of evolution. Darwin saw this clearly, and he was right to see it. But it is incomplete. And what endosymbiosis reveals is the nature of that incompleteness. The single most important event in the evolution of complex life was not a competition. It was a partnership. The most consequential innovation in the history of biology, the eukaryotic cell itself, was not built by one organism out-competing another. It was built by two organisms merging. By a predator that failed to digest its prey. By a prisoner that became indispensable. By two very different forms of life that, over deep time, learned to depend on each other so completely that they became a single new thing. Competition selects. Cooperation creates. Darwin knew this, even if his followers sometimes forgot. He wrote about the tangled bank at the end of On the Origin of Species. Organisms dependent on each other in complex ways, not simply fighting for survival, but existing in intricate networks of relationship. Lynn Margulis took that insight and ran with it further than anyone else had dared. She was difficult. She picked fights she didn't need to pick, and defended ideas she probably should have let go. She questioned the role of HIV in AIDS, which was wrong. She proposed spirochete origins for flagella, which remains unsupported. She was not a saint, and would have been the first to say so. She was a scientist, which means she was a human being, brilliant and flawed, and driven by something deeper than the desire to be comfortable. But on the central question of her life's work, the question of how complex cells originated, whether symbiosis played a fundamental role in the evolution of all complex life on Earth, she was right. Absolutely, unequivocally, triumphantly right. And it took the scientific world decades to accept what she had seen as a 28-year-old junior professor with 2 kids and no tenure. In the last interview published before her death in Discover magazine, the interviewer asked if she ever got tired of people calling her ideas controversial. Her answer: "I don't consider my ideas controversial. I consider them right." You are listening to this with a brain powered by the descendants of ancient bacteria. You are processing these words with mitochondria that carry an evolutionary lineage separate from your own. You are, in the most literal biological sense, a merger, a partnership, a collaboration 2 billion years old, still running, still working, still keeping you alive with every breath you take. The next time you take a deep breath, really take one, feel the oxygen fill your lungs, consider where it goes. Through your blood to every cell in your body. And there, inside those cells, it is received by mitochondria, by the descendants of that ancient alphaproteobacterium that was swallowed and never released. They take your oxygen. They make your energy. They keep you alive. They have been doing this for 2 billion years. We think of ourselves as individuals, separate, autonomous, alone. But at the most fundamental level of our biology, we are collaborations. We began as partnerships. We exist as partnerships. Every complex cell on this planet is proof that the most creative force in evolution is not destruction. It's the decision to stay. This is Cosmic Canvas, where we find the partnerships that built the world. Until next time, remember that every breath you take is powered by an ancient collaboration. You were never alone. Not even close.